Mayer, Executive Director of the Cholangiocarcinoma Foundation. I would like to welcome you to today's webinar. The topic today is the role of CEACAM6 as a biomarker in cholangiocarcinoma. We are very pleased to have Dr. Flavio Rocha here with us to discuss his work. Dr. Rocha is a staff surgeon at the Virginia Mason Medical Center in Seattle, Washington. He was the 2012 recipient of the ASCO Young Investigator Award funded by CCF. His research interests include translational research in liver, bile duct, and pancreas cancers. If you have specific questions during the webinar, please enter them in the question box on the right of your screen, then I will ask them on your behalf. Speakers will not be able to answer any personal questions during the presentation, so please be sure that your questions are general in nature. Dr. Rocha, I now turn the floor over to you.
Well, thank you, Donna. Thank you, Rick. This is truly just a wonderful opportunity for me to talk with you and this group. As you know, I was very generously introduced and funded by the Cholangiocarcinoma Foundation coming out of my fellowship, and have been maintained that relationship with you, and more importantly with the patients that I've been able to have the privilege to take care of. So what we'll go over today is just a little bit about the research that I've been conducting for the past 4 years, actually, since fellowship here in New York and now here in Seattle. And I'll give you a brief overview of what has been done to date, and then a little bit about what directions we're taking in the future. So I think, as most of this, from my experience, this audience tends to be very sophisticated, and I think what we've come to realize as physicians and surgeons and oncologists treating bile duct cancer is that they're really not all the same. We think of the bile duct as being one long continuous tube of which the gallbladder sprouts out from, but what we've learned recently is that there really is a different molecular as well as clinical behavior between the different types of cancers. Taking the gallbladder out of it altogether, even within just the bile ducts themselves, we know that the intrahepatic bile duct cancer tends to behave differently than the extrahepatic bile ducts. And even within the extrahepatic bile ducts, the perihilar or Klatskin tumors typically are managed very differently than the distal extrahepatic bile duct tumors. Unfortunately, because the numbers from a societal perspective have been so low compared to more common cancers like colon or breast, cholangiocarcinoma tends to be lumped in together, and all these populations of patients are lumped in together in order to get enough statistical power to conduct any clinical trials. But the hope is to get more attention drawn to bile duct cancer and trying to actually bring together investigators and patients, much like the Cholangiocarcinoma Foundation is currently doing, in order to have more dedicated research into the specific details. So again, as we all know, part of the challenge in this disease is that it tends to be a very difficult diagnostic dilemma. Typically, when this cancer presents, it can be very vague. Symptoms like abdominal pain, maybe some anorexia and malaise are very nonspecific. By the time jaundice and weight loss are present, it typically means that the disease is in somewhat of a more advanced state. And it may be a case where some of the treatments may not be applicable. Even looking at laboratory investigations like liver function tests, bilirubin, and so forth, will typically show an increase or abnormality, but these abnormalities can also be present in benign disease, just like hepatitis or pancreatitis. Part of the frustration is also there's no real perfect radiologic test. So X-rays, MRIs, and CAT scans can be suggestive of a tumor, but are typically not confirmatory. Adding to that frustration is that our current biopsy techniques may also not be very accurate. So as I explained to patients, unlike a breast cancer that you can feel and you can put a needle into, or a colon polyp that can be seen by endoscopy and removed, typically cancer of the bile ducts can be very insidious. They're not necessarily forming a mass. They could be crawling along the duct. And our best technique to date, which is endoscopic brushings, are only positive 40 to 70% of the time. Whereas I tell patients, essentially it's the flip of a coin. And so I never like to rely on the brushings themselves, because a negative biopsy is not necessarily reassuring. Only a positive one is, but we only get a positive biopsy half the time. Therefore, this is actually an area where biomarkers would be critical. Certainly, biomarkers for early diagnosis would be very helpful, and this is where I focused my research. From what we know so far, it seems that the serum CA19, or carbohydrate antigen 19-9, is the most widely used marker for both bile duct cancer, but also pancreas cancer. For those of you who don't know, it actually consists of an antibody that's raised against a colorectal cancer that reacts with certain epitopes on the cell surface. What's also frustrating at times is that it's not present in typically 7 to 10% of the population. So even though the marker is negative, that sometimes is not reassuring. Of course, if it's positive, it can be helpful. The other problem with the CA19-9 is it can also be elevated in the presence of obstruction of the bile duct. And so even with benign disease, we can see the marker go up. And therefore, it can't be used exclusively as a measure of cancer. In addition, just like CEA, the levels can be increased in smokers. Now, what about looking at biomarkers in bile? Certainly, the serum is very readily accessible. And the serum is the fluid component of the blood. You can get blood drawn through a peripheral IV, through an arm, a leg, and so forth. But it also contains a lot of other proteins that are involved in it. Bile, in this case, the advantage of bile is that it's really the most proximal fluid source, meaning that if we're talking about bile duct cancer, the likelihood of finding bile duct cancer proteins, bile duct cancer proteins would be in the bile, which is the most proximal source. The downside is that unlike the serum, which can be drawn by an IV, the collection of bile can be challenging, because it typically will require an invasive procedure such as endoscopy, or for patients undergoing a surgery, an operation for something, they can have bile collected at that time. The other challenge with bile is that it also needs to be purified, and other things need to be taken into account, because bile not only contains digestive enzymes, but bile salts, cholesterol, and other factors as well. But let's review. Has this been done? Well, if we look at some of the older series here, looking at analysis of proteins from bile, particularly for cancer diagnosis, we can see this is a summary of 4 studies looking at how many proteins were identified. If you follow my mouse, you can see that there can be up to 109 or 117 different proteins that were identified that were in common, and even 70 or 53 different proteins that can be found. This is kind of an amazing phenomenon, given that proteins only make up 7% of all the content found in bile. Well, what is the difference between serum and bile biomarkers in cholangiocarcinoma? This was a nice study published out of Italy, looking at serum markers as well as biomarkers. And you see that the ones that are commonly used, like CA19-9, has a pretty broad sensitivity and specificity, or what is the likelihood that this will be picked up on the test, and what is the confirmatory capacity of that marker. And you can see it's a very broad range. The same thing with CEA, a very broad range in the serum, although it tends to be a little more specific, meaning that if you have it, you're more likely to have the disease. Let's switch over to bile, and at least in this study, looking at CA19-9 in the bile did not seem to improve either the sensitivity or the specificity. The same thing with CEA. However, this group looked at a different protein called insulin-like growth factor, and they measured this in the bile and in the serum of patients undergoing ERCP, or the endoscopy to look at the bile ducts. And in this population, they were able to find that in the serum, there was really no difference in IGF-1 between cholangiocarcinoma patients or pancreatic cancer patients or patients that had benign disease. But when they switched over to the bile levels, you can see that now they were able to pick up the IGF-1 levels in cholangiocarcinoma patients much higher than they would see in pancreatic cancer or in benign disease. So this led us to look at CEACAM6 as a potential biomarker for cholangiocarcinoma, much like IGF-1. So what is CEACAM6? Well, as you can see from the name, it is actually a member of the CEA family, so the same tumor marker that we've used for cholangiocarcinoma as well as colon cancer. The difference about CEACAM6 is that if you see in this diagram of a cell membrane, the CEACAM6 is only found on the surface of the cell. So it does not have any intracellular domains. You can almost think of it as having little hairs on the outside of the cell. What does it do? Well, it has several known functions. So we know that it actually serves as a cell adhesion molecule, so that cells can either stick together or stick to the walls of tissue. And it can actually transduce signals, meaning that if something is attached to it, it can actually transmit the message inside the cell. We know that in cancer cells, it actually can modulate the behavior of these cells and make them either invade or resist killing by chemotherapy, or anoikis. Anoikis is an interesting phenomenon where our cells tend to clump together, and that's how they live. But when the cells are usually detached from tissue or from themselves, they tend to die because they're not supposed to live by themselves. But a phenomenon that occurs in cancer cells is that they're able to split off from their home, as most of you know, and then travel to different sites. And CEACAM6 allows that cell to live independently and then spread. We know that CEACAM6 is actually present in several other tumors besides colon and cholangiocarcinoma. It tends to be present in lung and breast cancer as well. And when present, it can be associated with a worse outcome in these tumor types. When I was a resident in Boston, our laboratory was very interested in CEACAM6 as it applied to pancreatic cancer. So this was a study published by my colleagues in the lab almost 10 years ago now. And interestingly, in pancreatic cancer, CEACAM6 was expressed in almost greater than 90% of the tumors. And you can see here in the panels, this is what a normal pancreas looks like in panel A. Panel B has a pancreatic cancer that does not express CEACAM6. And in panel C is a pancreatic cancer that does express CEACAM6. And you see from the survival curve that the majority of tumors did express CEACAM6, and the survival was much worse in this population. And these were patients that both underwent resection. But if the tumor did not express CEACAM6, the survival was better. These findings were also at the same time discovered by a group at the time at Johns Hopkins, and they were looking at the pancreatic juice. So much like the bile duct is the most proximal fluid to bile duct cancer, pancreatic juice is the most proximal fluid to pancreatic cancer. And they looked at several proteins, and interestingly, they had CEACAM6 as part of the proteins they looked at. And according to their series, there was a 182-degree fold increase of expression in the juice of pancreatic cancer. So we know that it was being shed. A similar group from France this time looked at bile, which is the fluid of my interest, looking at the levels of CEACAM6 and CA19-9 in patients with both pancreatic cancer, pancreatitis, which is benign, gallstones, also benign, and they had a small cohort of cholangiocarcinoma patients. Now, if you're not used to seeing these blots, this is what's called a Western blot. And what a Western blot is that you take the proteins from the fluid, in this case bile, and you run them on a column or a gel that separates the proteins by different weights. So depending on how heavy it is, it will actually not make it down as far. So it'll be the kilodalton weight of the protein is what makes the difference. And then you take that gel and you expose it to antibodies to different proteins. In this case, CEACAM6. as well as CA19-9. The numbers here represent different patients. And so what you'll see here is that there were 2 patients with pancreatitis, and when the CEACAM6 antibody was used, you don't see any reaction. So there was really no CEACAM6 present in the pancreatitis patients. However, when you look at the patients with pancreatic cancer, you can see that most of them, all but one, had very dense expression of CEACAM6. So indeed, it was present in the bile of these patients. Again, moving on to gallstones, both the 2 gallstone patients did not have any CEACAM6 expression, but the 3 cholangiocarcinoma patients did, 2 more than this patient number 12. By comparison, when this group looked at CA19-9 in the bile, again, there was none found in pancreatitis. There was some, but to a lesser degree, found in the pancreatic cancer patients, none in the gallstone population, and probably at least 1 out of 3 that you could see in the cholangiocarcinoma patients. The group from Japan looked at CEACAM6 gene expression, this time not in the bile, but in the actual tissue of cholangiocarcinoma. And this was actually a specific subset of intrahepatic cholangiocarcinoma patients. Now, by gene expression, they were looking at the actual message. So we know from our biology class that the way proteins get made is that there's a genetic code in the nucleus that gets transcribed. That transcript is then exported out to the cell, which actually assembles the protein. And so this was looking at that code, or that transcript, from both normal tissue and tumor tissue. And what they were able to find is that this message of CEACAM6 in the cancer tissue was much more elevated than in the non-cancerous tissue. Furthermore, in patients that did have cancer and had their operation, those that had a high expression of CEACAM6, you can see had a worse survival than those with low expression of this transcript. So given this information, when I was a fellow at Sloan Kettering in New York, that has a large bank of tissues of cholangiocarcinoma, we decided to do a pilot study. And this is where, again, I am indebted to the Cholangiocarcinoma Foundation for allowing us to perform these studies. And so we made what's called a tissue microarray. And what a microarray is we take tumor types from several patients, hundreds of patients, and we take a small little bite and put them all on one slide. So as opposed to having just one tissue per slide, we have 100 tissues per one slide. And these can then be stained for different proteins, in this case, CEACAM6. And then the pathologist can score the staining. And then with the clinical information from each individual patient, we can then start to make some associations between the presence of CEACAM6 and any of those clinical or pathologic variables, including survival. So this is an example of what these cores look like. And again, you'll see this is very similar to the other pancreatic cancer study. So this is what a normal bile duct would look like. This is a bile duct cancer that does not express CEACAM6. And this is a positive control of a CEACAM6 expressing tumor. So you can see that it's typically not so subtle, the expression. Well, what did we find? Well, in this cohort from New York, we had 120 patients, of which 60, the majority, had perihilar or Klatskin type tumors at the bile duct bifurcation. There were 45 patients with intrahepatic cholangiocarcinoma, and then 15 patients with distal cholangiocarcinoma. You can see here the distribution of age, sex, the type of resection, the differentiation, lymph nodes, stage, and then here is the expression, the proportion of those that had CEACAM6. And you can see that in the hilar cholangiocarcinoma patients, almost half of them expressed the CEACAM6 protein. That was the same for the distal cholangiocarcinoma patients, roughly half. However, the intrahepatic patients, it was actually the minority, so only 16% of those patients expressed CEACAM6. how did the CEACAM6 positive tumors compare to the CEACAM6 negative ones? Well, what we're able to tell from this study is that they tended to be somewhat older, a small percentage to be older, not big difference in sex, not a difference in resection or differentiation. There was a trend towards patients having CEACAM6 not having a papillary morphology. And this is important because papillary morphology tends to be one of the favorable categories in cholangiocarcinoma, meaning that papillary tumors tend to be somewhat less aggressive. So again, this fits with the notion that a CEACAM6 positive tumor tends to be more aggressive. And you can see that there was also a significant increase in the higher T stage of patients that presented with CEACAM6 tumors. We then looked at disease-specific survival in each cholangiocarcinoma subtype following resection with negative margins and stratified them by CEACAM6 expression. Again, with that notion that not all cholangiocarcinomas are the same. What were we able to find? Well, in the cholangiocarcinoma patients, remember, these are, again, 50% of those had CEACAM6 tumors. But when we plotted out the survival, there was really no difference whether the patients had CEACAM6 positive tumors or negative tumors. Now, in contrast, the distal bile duct cancer patients, again, there was a very small number of patients that was there. And this was interesting. This was actually, this wasn't statistically significant, but it appeared that, again, CEACAM6 may not have had as much of a difference, but these are small numbers. Lastly, though, the intrahepatic patients, there was a stark difference. And as you can see that the significance value here is highly significant. That patients that did not have expression had a much better survival than those that had. Now, again, this was a small minority of patients in this scenario, but you can see that if your tumor had CEACAM6 present, even if you underwent resection with negative margins, the survival was poor. But what did this mean? Well, it means that there is something about CEACAM6 in the cholangiocarcinoma that makes a difference. Probably one of my proudest moments is when I left New York, that based on this data that we had collected, my mentor, Bill Jarnigan, who was the chief of hepatobiliary surgery at Memorial Sloan Kettering, actually incorporated this information into a clinical trial that's actually currently ongoing through clinicaltrials.gov at Sloan Kettering. This is actually the trial number. And what this is Memorial has had a long tradition of giving intrahepatic artery infusion of therapy to patients with unresectable cancers. And this tradition started with colorectal cancer, where they were able to achieve dramatic responses. And what they've done is they've taken this approach and applied it to patients with intrahepatic cholangiocarcinoma. And the combination of chemotherapy through the hepatic artery, as well as the systemic component with gemcitabine and oxaliplatin, they've actually are now accruing patients to this trial. And the idea here is that we're going to try to see if there is a difference at the time patients get the pump placed. They get a liver biopsy to see the CEACAM6 expression. And we'll see if this treatment has any effect on those CEACAM6 positive tumors that have that worse prognosis. So as you can see, this is an example where just basic lab and translational science can then be rapidly applied to clinical trials and hopefully eventually to clinical care. So as I left Memorial and made my way to Seattle, we wanted to take this research further, and we began a protocol to prospectively now collect the bile from all patients, both at Sloan Kettering and at Virginia Mason, that were undergoing resection or bypass procedures for bile duct cancer, gallbladder cancer, pancreatic cancer. And also, more importantly, since Sloan Kettering is a cancer center, we never had any good controls to collect bile from patients with normal disease. And so as I got here to Seattle, we started collecting bile for patients undergoing either surgery or CP for benign disease, just like pancreatitis or gallstones. Because bile is abundant, we were able to collect up to 5 cc's either during surgery or CP, And these were snap frozen in liquid nitrogen. And then we looked at the CEACAM6 levels using those same techniques that I had described. We then try to correlate the findings of CEACAM6 based on the patient's diagnosis and their outcome. Again, with the support of the foundation, we were able to publish these findings actually last month. This is the title of the paper, which is available, and you can see on the bottom there, through PLOS One, which is a public library of science. And this is an open access journal, so anyone has access to them, to the information. And you can pick up, and I will show some of the data from this paper that, again, is immediately available. You can see here the cohort of patients that we had. So we had 49 patients with benign disease, and then 24 patients with extrahepatic, and 18 with intrahepatic disease. This is the percent that were male, the percent that were stented, and the percent that had, the bile salt concentration, the CA19 level, the CEA, alkaline phosphates, and bilirubin in these patients. We then quantified also the CEACAM6 levels, and the ELISA is another form of measuring proteins. So it's an enzyme immunosorbent assay. And this is done sort of through a rapid throughput where we can actually take a plate that's coated with the antibodies for CEACAM6, and we can just drop with a pipette the from different patients, and we can then get a readout of the levels. And what you can see is that in patients with benign disease, the CEACAM6 level is quite low. It's only about 8 nanograms. But when you have patients with cholangiocarcinoma, that goes up to about 40, and this is in the entire cohort. This was statistically significant. When we break them down between intrahepatic and extrahepatic, certainly the intrahepatic patients had a smaller level, where the extrahepatic patients had a much higher level of CEACAM6. The way we measure correlation between levels and disease is what's called a receiver operator curve. And what this does is it plots the sensitivity versus the specificity of any given test. And so the closer you are to 1.0, the best the test is. Anything close to this line, which would be an AUC of 0.5, means that it's a coin toss. And what you can see here is that in the entire cohort of populations, the area under the curve, which is underneath this line, was about 0.74, which is very respectable. However, there was a difference between intrahepatic and extrahepatic disease. So that curve was much better, again, approaching 1 for extrahepatic disease, as opposed to the intrahepatic cholangiocarcinoma. We again looked at the Western blot, and this you should be familiar with now. And this is just a representative one between patients that had benign disease and those with cholangiocarcinoma. And this is, again, the marker for CEACAM6. And you can see, again, that there was a very dense expression of the marker. And so in summary, the research has shown that CEACAM6 truly is a prognostic marker, both by message and protein, in the tissue of intrahepatic cholangiocarcinoma. The second part of this is that CEACAM6 levels in bile can be used to distinguish cholangiocarcinoma, but probably in the extrahepatic disease and not intrahepatic disease. But you can't tell them between cancer and biliary disease, and benign biliary disease. What's left to do now is really to determine if CEACAM6 can be a predictive biomarker prospectively through clinical trials and more translational research. Let me tell you where we're going now and what the focus of my lab is currently. So CEACAM6 is a protein. What we started to focus on now a little bit while the CEACAM6 clinical data is matured is looking at what's called MicroRNA. And you may ask, what is MicroRNA? Well, RNA is, again, part of that message that gets transcribed from your DNA, which is your genetic code. And most RNA is used as a code. Now, microRNAs are really, as the name describes, small little pieces of genes that are then exported out of the cell nucleus and are then unwound, translated, and made into other messenger RNAs. They've actually only been recently described, but as you look here on this table, this is microRNAs from different cholangiocarcinomas, and you can see here whether it's extrahepatic or intrahepatic. They have several numbers associated with them, and you can see here that their expression can be altered depending on what type of message they are. So in this scenario here, this microRNA 320 is downregulated. This is its target protein that gets transcribed. So you can see that there is a pattern, or if you will, a signature potentially. So what we want to do is look at microRNAs in bile. And so we took our bile that we have collected from patients, and we looked at bile either from the gallbladder or from the bile duct. And you can see here the different locations. Oops, let me switch back. Whether that was done from the bile in the liver, in the hilum, or in the distal bile duct. And what we're able to tell is that the quality of the microRNA in the gallbladder was degraded. And the same thing as if the bile was obstructed. And so we couldn't get as good of a harvest. But if bile was collected from a bile duct absent of obstruction, we could really get some information. And you can see here, for example, the difference between 3 separate patients, one of whom who was obstructed, others who had, this one was not obstructed, this one had a moderate level of obstruction. And you can see that the yield of RNA is much higher depending on the level of obstruction, whether it's from the bile duct or the gallbladder. And so we wanted to see as well, is there a change in the microRNA expression as cholangiocarcinoma is developing? Because again, part of the problem with this disease is that we're getting to it too late. Is there a way to find out about the microRNA signature as the cancer is developing? This would certainly be very relevant in patients who we know are predisposed for cholangiocarcinoma. For example, the primary sclerosing cholangitis patients, or PSC, that typically tend to be associated with inflammatory bowel disease. But again, we don't know when's the right time to intervene. So what we try to do is we then took this information and went back to the lab to an animal model. And so we took rats and we induced cholangiocarcinoma in them. So there was a model of giving them a drug called thioacetamide. Over time, these animals actually develop intrahepatic cholangiocarcinoma. And what we're able to do is then at different time points, after we expose the animal to different to the drug, we were able to sacrifice at certain intervals. So we sacrificed them at the beginning, at 4 weeks, at 10 weeks, 16 weeks, and by 22 weeks, almost all the animals have confirmed tumor. We collected serum, bile, and tissue from these patients, and from these rats, and then analyzed them for microRNA. You can see here what a rat cancer looks like. So this is a normal rat liver, and then this is a rat liver with a big tumor sitting in it. What we got out of this information was a lot of endpoints, and so we actually enlisted the help of some of the computational biologists, and we had what's called the Volcano plot. And what a Volcano plot looks like is you can see the different levels of expression. And so this is the 0 mark, and microRNAs that are expressed at a different fold, so 1-fold, 2-fold, 3-fold, 4-fold, are plotted on the right, and then those that are downregulated by a certain fold, -1, -2, are expressed on the left. And you can see here, for example, what a control animal looks like and what that microRNA pattern changes over time. What we found in our tumoral microRNA is you can see here, this is now a plot looking at, this would be the 0-fold expression. And you can see the number of microRNAs that are now we were able to discover that are being either upregulated or downregulated. And we can see the fold that they were upregulated. And what this is now is a roadmap, potentially, to study those different microRNAs. And so this is where our lab has focused attention currently, and we're going to try to find if there's a signature at a certain time. If you see this microRNA going up, we are heading towards potentially a cancer developing. So what I'd like to do at this point is acknowledge my colleagues, both at Sloan Kettering, my mentor Bill Jarnigan, and certainly the other surgeons at Sloan Kettering, the pathologists that worked very closely with us to actually stain the tissue, and the biostatisticians there. Here at Virginia Mason in Seattle, give a shout out to Bart Rose, who worked in my lab for 2 years, helping us out with this project, my hepatology colleagues, as well as our pathologist here. I'd also like to acknowledge the Conquer Cancer Foundation of ASCO that really provided the seed funding for this, and this was actually funded through the Cholangiocarcinoma Foundation, who I've had the wonderful experience to be in touch with and is hosting this webinar. I've subsequently was able to retain more funding through our Research Pioneer Award and to continue on these studies. And lastly, I really wanted to acknowledge my patients and all the patients with cholangiocarcinoma and their bravery to actually go through this treatment and be interested in trying to figure out how we can get to this disease and make some improvements. That's all I had planned for today, but I am happy to take any questions for those present. Again, thank you very much.
Thank you, Dr. Rocha. That was excellent. We do have a few questions. The first one is, what causes the differences in the 3 different types of cholangiocarcinoma? Is it location only?
Yeah, that's an excellent question. You know, it's hard to know, and you know, CEACAM6 is not the only protein that's differentially expressed. If you look, Andrew Zhu from Mass General put out a nice paper in Journal of Clinical Oncology looking at other important proteins like KRAS, more recently IDH, which we know is another very important protein, and for some reason they are differentially expressed. Is it location itself? Probably not, but I think there's something about the milieu, whether it's within the liver or outside the liver, that may have some predisposition for some proteins to be more active than others. But that's an active area investigation, and I think part of the issue with treating cholangiocarcinoma is that, you know, we tend to just lump them all the same way. So, you know, if we have a new targeted therapy that may work on a specific protein, we end up, you know, in the past we've exposed every patient to them, and some patients may not benefit from it. We really have to, you know, take cancer treatment to a more tailored approach, or so-called personalized medicine. And so I think the more we're able to learn about not only the location of the tumor, but the individual DNA signature of that tumor, then I think the more we can help patients. You know, what I tell my patients is that, you know, your tumor is like your thumbprint. It's got your DNA on it. So even though it may be the same quote-unquote site as, you know, somebody else, it really is your own signature.
Okay. The next question is about the biliary brushings. You mentioned that the statistics show that they're unreliable. Why is that?
So, yeah, part of the issue, and this is sometimes very frustrating, I think, to explain, is that bile duct cancer, even though it's on the, you know, the surface, the luminal surface of it, tends to spread submucosally, or so-called under the lining, and it spreads up and down the bile duct and also out. If you can think of it, it's sort of in 3 dimensions. So unlike a ball of cancer, so you think of a breast cancer as being a ball that you can stick a needle in, the bile duct, you're really trying to scrape it. So you're trying to scrape cells off a surface, and you may, 1, damage the cells so that they can't be read, and 2, the majority of the tumor may have already slid underneath the surface and is crawling as opposed to being on the surface. So that's been the major challenge. You know, and our colleagues in gastroenterology have come up with other, you know, techniques that you may have heard of, such as spyglass or confocal microscopy. And, you know, those have maybe increased our ability to biopsy lesions better because you can target the biopsy. Remember, these brushings, when they're being done by ERCP, are essentially being done blind. The gastroenterologist has the scope in there. They just take the brush in there and they kind of scrape. The spyglass allows you to at least focus your biopsy to a location that looks mostly abnormal. But as I tell my patients, if I see somebody that's got a biliary stricture, I sort of take that, that for no reason, meaning that they didn't have a bile duct injury, didn't have anything else done. I take the fact that to me it's a cancer until proven otherwise. And so if a biopsy is positive, it's unfortunate, but it helps you. But if it's negative, I tell them that it's I don't want them to be falsely reassured. So we have to then move on to something else.
Okay. Just as a follow-up to that, you mentioned bile duct injury. Is that with the primary sclerosis?
No.
PSC, or what would cause a bile duct injury?
Yeah, sorry. So part of what I do is, as a surgeon, is that I fix bile duct injuries that have happened to patients who've had their gallbladder out, for example, and they've, unfortunately, the main bile duct was injured at that time, and those patients can develop strictures, but they're typically due to inflammation or scar. So that's a separate population. The PSC patients can develop strictures from the inflammatory process of their autoimmune disease. And what we know is that having PSC is then a risk factor for developing cholangiocarcinoma in the future from that inflammation, that continued inflammation.
Mm-hmm. And what about other autoimmune diseases like Crohn's or colitis?
Yeah, so we know that PSC is associated with ulcerative colitis. And so those patients are typically the ones that develop the PSC-related cholangiocarcinoma. And those are the tough ones, because a lot of times those are very young patients that have had their colon out for the UC, but they still may be at risk of developing PSC in the future.
Okay, we have a question from a patient in Montana. Wants to know if they can request the CEACAM6 test. Is it available in all areas?
That's a great question. No,
know how to get it.
Yeah, it's not quite clinically available, because this research has just kind of come to fruition in the last couple of years. So, even though, we have kits available for laboratory investigation, it is not a clinically available test yet. I'm hoping to change that. Okay,
but if the patient wanted to have this test, is that something that they would request from a surgeon or from their doctor, or that's not even an option for them?
No, because obviously there's a lot of regulatory things that go into effect for diagnostic tests. It's not currently FDA approved, so you'd have to have approval from a regulatory agency to use that for diagnostic purposes.
Okay, and then the last question we have is about the thioacidamide. So is that something that humans are also that you've injected rats with that? Is that something that humans are also exposed to?
Gosh, I hope not. I get this asked. It's a very good question, because I've been asked from, I have patients that work in shipyards and are mechanics that ask, because it's such a rare cancer, is there a reason why, some patients may have it or not? And environmental exposure certainly can be a risk factor for cancer. As far as I know, there's not anything that thioacidamide is in that we normally would ingest or use. It's really just a chemical that has been researched, and because it causes cancer in animals, has been adapted to serve as a cancer model. But no, not that we know of.
Okay, that's reassuring.
Yeah.
So that was the last question. I'd just like to say on behalf of the Cholangiocarcinoma Foundation, we'd like to thank you, Dr. Rocha, for sharing your time and expertise. For those who are participating in the webinar today, I want to let you know that we will be posting the recorded webinar and the PowerPoint presentation on the CCF website. If you have additional questions, you can post them on the CCF discussion board, and we will make sure that you receive a response from Dr. Rocha. Thank you to all who participated today. The webinar will end now.
Thank you very much.